Thank you so much. Okay, I am in the Spanish channel. So it's a pleasure to introduce Guillermina. As you know, Guillermina, a few years ago, I met her when she gave a talk at the lab where she did her postdoc. And so I've been following sort of her career and her research since then. She did her undergrad in physics at UNAM, which is the largest university in Mexico, the largest public university. She got her doctorate, her doctor's degree in biophysics under Margaret Gardner at University of Chicago. And when I met her, by then there was this interest in cellular migration. So the idea of this migration in extracellular environments. And then she worked on the role of cytoskeletic patterns in the morphogenesis of organs. So that's when I met her. And she did her postdoc at UCSF with Manu Prakash, which is where she was studying fluid mechanics in cellular biology and celiac arrays. So this was one of the subjects I was most interested in, how the cilia were organized and configured to clear the lungs. So if this is something of interest to you, I would encourage you to put your name in the chat. For those who aren't aware, Guillermina's very involved in scientific outreach and does a lot of work with Manu Prakash that are known at an international level, and it's done through open hardware. So, it's low cost, it can be taken out into the field. And it's this idea of the importance of access, technology access. She's now at EPFL in Switzerland, studying biological patterns and functions within molecular interactions at a microscopic level. And so the flow mechanics with active filaments or cilia and extreme cell mechanics. And I will let Guillermina speak. Thank you so much, Mariana, for that introduction. And I apologize. It always makes me feel so bad that I have to discuss my research in English. But the thing is, I never learned biology in Spanish, so I do have to give this talk in English. But I hope you can enjoy it anyway.
As Mariana said in her kind introduction, my group now is really interested in studying living matter, and particularly from the lens of physics. So we study biological systems and we do a lot of microscopy. So here you can see movies that are actually data from living organisms. And in this case, we have a lot of things that have cilia. And this is basically what we do. We do a lot of imaging, and then we try to apply kind of the lens of physics and understand biological systems from a different perspective. So what we really want to do is use this lens of physics where we really think about molecules all the way to the macroscopic scale and try to integrate the function of things, right? We can think about in biology, the smallest things are molecules. These are nanometer scale assemblies of proteins that already interact. There's a lot of things going on. And somehow all of that gets integrated into a cell. Here you can see a cell that is migrating. It has organelles. So this is the micrometer scale. And already you have that cells we can think have motility, have many functions that are all coming from the molecular scale. And then, of course, you have tissues. This could be closer to the centimeter scale. and finally, organisms and the environment. So here you can see a cute picture of my daughter as an example. This was a year ago. But you can see that ultimately, all of these things come from the action of proteins. So then if we want to understand how an organism works, it's really hard if we think about this question. So from the lens of physics, what we do is actually abstract all of this complexity at the molecular scale and just try to understand the system from the mesoscale and try to see, okay, can we actually do some measurements in this microscopy data that map into some biophysical parameter space, and then we can understand the behavior. And you can see already that this is a beautiful problem. Here are just some examples at these different length scales of patterns that you see across biological systems. So already at the nanometer scale, here what you're seeing is a basal body in the left. And then you see this beautiful structure that has symmetry, that then each one of these anchors a cilium to a cell. Then you see these rows of microtubules, and each one of these little dots is one of the structures that I just described. So you get an idea of actually how things organize, then organize into a bigger length scale, into the patterns that, for example, we see at the famous flocks of birds in Rome. So you can see that at every length scale, you have rich phenomenology, lots of complexity, and really the challenge is how to understand this. So this is the point of view that we take in my group. And as you can already guess, this requires integrating expertise from lots of different disciplines to really try to understand what's going on with biological systems. So things that we think about in my group, for example, are how are patterns formed? So this is a question about symmetry breaking. So this is statistical mechanics. How are they maintained? How are they evolving? And then we are really interested as well in connecting the physics, not only the physics, but also understanding the biology. So for this, we really need to understand the molecular basis of the patterns we're seeing. So we draw a lot from physiology, development, cell biology, and then in the end, a lot of these things exhibit self-organization. So then we can think of them as matter as well. And then ask about from a material science perspective, what are the properties that are actually that this biological matter has that gives rise to the emergent behaviors we see. So this is the idea in abstract. But then just to give you a better idea and a clear example from physics and how do we integrate knowledge across these organizations, we can think about, for example, forecasting weather or large patterns that we see in the atmosphere. So if you really think about it, like we look at Earth, we have a little piece of atmosphere. So what is in the atmosphere? We have some water molecules, we have oxygen, other things. And then here you can see these little cubes, each one is a molecule. But then if we want to understand weather patterns, like for example, these ripples that we see in the atmosphere, then it's really not helpful to try and look at the physics all the way from the molecules and then kind of run the simulation and see if we get these patterns. So instead, what people do is this mesoscale analysis where we think of things about in the framework of continuum mechanics, for example. So instead of thinking about every individual molecule in here, we just think about a piece of material. And then instead of the cube, we just think about the cube, not what's inside. And then try to abstract everything that's going inside via understanding what are the forces that are acting on this piece of material or of atmosphere in this case. And then from this, we can arrive at an equation, for example, in this case, the Cauchy momentum equation that will describe actually accurately all these patterns that we see, for example, here on the right. And this is an approach that has been successfully applied in biology, especially in this field of development, pattern formation, cytoskeletal mechanics. And here you can see that this same equation that predicts atmospheric patterns can actually predict the looping of the gut. So then we have seen that this is a successful approach, but then of course we always need to remain grounded in the biology and think about ultimately what are the molecular underpinnings of what the forces that go on in this little cube. But this is just to give you an idea of how we apply this approach in general and specifically to biology, as Mariana mentioned. This has kind of been a lot of the basis for the work I've done. First, during my PhD, I did some work trying to understand tissue shape changes in the context of Drosophila chambers and how they become elongated. And then so there's a lot of things going on, but then it turns out if you actually measure the alignment of some cytoskeletal filaments, then you can see that you can actually predict how this thing is going to arrive at its final shape. Similarly for cell motility, rather than thinking about, okay, each molecule that could be important for cell migration, if we actually screen through a bunch of them, we can see that really these molecular perturbations map into a space that has to do with physical parameters. For example, the area of a protrusion and how it is oriented in space. And then you can have a kind of, okay, these two parameters that control the behavior of the cell in this context. And then today I will talk more about, as Mariana mentioned, a more recent work that I've done understanding how flows are generating. So then this will be the focus of this talk. So here it's also good for our interpreters. I want us to pause and take a deep breath. Right, so if we pause, we took a deep breath. I always like to imagine that this is what's happening in our airway. So this is actually a microscopy from some particles being affected by the multiciliated epithelium in a mouse airway. So then, as you can see, this group of cells generates fluid flows that are highly coherent and well organized over the centimeter scale. And then this is actually mucus clearance. This is also what happens in our lungs. And then we have this epithelium that lines our airway. Then we have a mucus sheet that gets transported with all the particles onto the outside. So of course, how you engineer this flow is very important. So we try to think about how we would build a flow like this if the elements that we are using to build it are these cilia. So cilia are these long, slender organelles that are attached to cell surfaces. And then they look exactly like this, like a filament. And this filament beats at 20 hertz or so. And then this actually generates the flow that we see, but then we run again into this scale disconnect. So each cilium is around 10 microns in length. And somehow they produce the centimeter flows. And here you can see actually the pattern of beat of each cilium. And this is what generates this flow proportion. But if we want to understand how you go from here to here, then you have to think about how cilia are organized inside of cells. So each cell in this epithelial layer has hundreds of cilia. And then, of course, you have hundreds of these cells that together generate the forces that drive this highly organized flow. So then the first question that we try to think about in this context is how do you build a flow if you think about the actual components that you have. And then here the problem really biologically speaking is that this flow needs to be very organized, highly directed, and very robust, right? Because as we mentioned, this is a very important physiological function. So we have ciliated cells here in green in this cartoon. So then I know that I have a few of these cells. And then I have this whole airway, and then I have to generate a flow that goes from the lungs to the mouth. And then, so how would I organize them? So first, I know that the first constraint that I have is that I cannot have a whole carpet of these ciliated cells because we need other cells in this tissue, for example, to secrete mucus or for other immune functions. So then because of this, we will have a coverage fraction of the airway less than 1. So not all of the available surface of the airway is going to be covered by the cells. So then we can think about, okay, then it's very important how we actually organize them here. So how would we actually describe the organization of cells? So for this, we came up with this parameter that is called the wavelength, which is just the average distance between clusters of cells. And here you can see that, yes, so if we think about the cells, they have to be spaced somehow such that everything travels fully from one side to the next. And then what I haven't mentioned yet is that each one of these cilia that beats has a polarity, so it beats on a plane, right? So then this is what we will describe as the orientation. And then somehow cilia within a single cell must coordinate such that they don't collide. And then also across cells in the tissue, we will need to see what is the orientation of the cilia in one cell compared to the other. And then presumably that would be important for the actual architecture of the flow that is being generated. So what we did was did a lot of microscopy. And then, as I said, we like to think about this problem at different length scales. So then we started imaging from the smallest organization to the largest. And in this case, if we really zoom in into each one of these cells in the airway, you can see these little dots. So these dots are the anchoring points of each cilium. And this has an asymmetry. So there's two proteins that actually, if you look at their localization, they form an axis where you basically connect the dots. And that tells you the plane of beating of each cilium. And then here, what you can see are arrows. So this is immunofluorescence microscopy, and each little green and red dot is an individual cilium, and then you connect the dots, and then that tells you the direction in which each cilium beats. And if we zoom out, we can see here, these are in blue, the outlines of each cell, and you can already tell that each cell has hundreds of cilia. And then they are not perfectly parallel, as you can see in these arrows. These are actual measurements that we did. And if we measure the order parameter, which is something that tells us how parallel each arrow is with respect to the other, if they're all parallel, this will be 1. If they're all disorganized and completely, they have random orientation with respect to each other, this would be 0. So then you can see, we can think of this as a field, vector field, and then when we measure the orientation, we get that is something around 0.6. And as I mentioned, if they're perfectly parallel, they should be 1, but if not, it's less. So this is highly disorganized. Although it's not random, like I said, if it were random, it would be 0. There's still some clusters of cells of cilia that are organized, but not across the whole cell. And since we're thinking about maybe what happens at the mesoscale is what matters, because in the end, in this tissue, we have hundreds of these cells. Here, maybe we can abstract all of this and then just give each cell a vector that will indicate overall in which direction it pumps fluid. And then we can think about how cells across the tissue organize with respect to each other. And here you can see already from this polarity map that their cells are not perfectly parallel. And again, these are actual measurements from the microscopy. And we get, in this case, that the orientational order corresponds to 0.8. So then we buffer a little bit of this cilia level disorder, but still it's not 1 in this case. And what I want to mention here is that, for example, other systems where this organization has been measured, for example, in the case of the fly wings, where you have hairs, you do get perfect organizations, like where this order parameter is 1. So then we thought this was really surprising because in this airway, as I mentioned, it's really critical that you generate a flow that is directed in one direction. So then this was surprising. And another aspect to this disorder that we call is that here you can see microscopy zoomed out even further. In this case, each one of these little footprints is a cell. And then in green, you can see the cells that actually have cilia. So everything that is black in the background is actually cells where you don't have any activity. They're other cells. And if you measure the average distance between these clusters of green, you get that is around 16 microns, which is around 3 cells. So you have the cells that are really clustered together in a way, which is kind of what we suspected from how we introduced this. If you want to create a large scale flow, then you can't be too far away from your neighbor. But what was really surprising is that the coverage fraction, so the surface, if we zoom further out and now count how much of the total surface of this mouse trachea is covered by these multiciliated cells, it's around 0.4. So that means that less than half of the surface is cells that don't have activity. So in a way, it's really remarkable. You have other cells that don't have activity, yet you're able to build a macroscopic unidirectional flow. So then we ask, well, how could we understand this from a physical point of view? And what you can do is actually think of a model experiment. So then in this case, in collaboration with another postdoc at the time in the Prakash lab, Arnold Matthijssen, what he did is, okay, we can think of this tissue as this slab here that we see. And then what's nice there is that we can pattern any pattern of ciliated cells. We can pattern the activity and understand what will happen with the flow if we change the organization that we have of the ciliated cells. And we can do this in a very coarse-grained way, so we don't think about each individual cilium or not even about cilia at all. In the end, what we do is we just introduce a sleep velocity at the boundary where we have clusters of cells. So that's going to model the activity of the cilia. And then once we do that, then we're going to ask what type of flow we generate. And then, as I mentioned, we can vary different parameters of the architecture of the tissue that we're building. And today I will just tell you about one parameter that was kind of the most surprising for us. If we actually change how we orient the cilia with respect to each other in this case, or the actual direction of the velocity in the case of the model. So here you can see the simulation results. So each one of these green arrows represents a multiciliated cell. So then it's just gonna push fluid in a direction, and then the direction of the arrow is actually corresponding to the polarity of that cell. And then you can see if we can change the orientational order. So if we start at the left, then all of the cells are perfectly parallel to each other. All of the cilia point in the same direction. And then we start to introduce a little bit of noise to try to mimic the architecture that we saw in the cilia carpet. And because it's a simulation, we could just introduce as much noise as we want and change other patterns of the system. We can also change how many multiciliated cells we have, how far away they are. So we map the whole parameter space, but as I mentioned, I will just show you kind of the result of what happens if we orient cells differently. And then here you can see on the left that if you have some red areas where things are getting trapped, so there's recirculation, as you actually increase the disorder in the tissue, so we put here a value of 0.8, these areas where previously things were trapped are actually the streamlines are open, and then you can connect the dots and find a path to move out from there to the end. So here you can see those are the yellow arrows. But then, of course, as you introduce more disorder, things don't go anywhere. And we can measure this by calculating the clearance time and the total flux. So the total flux is just the total amount of fluid that goes through this little piece of tissue. And then you can see the more disorder I have, then the less stuff goes by. And the clearance time is, it's a mean exit time, but you can think about it as if I have this piece of tissue, I throw in some particles, then I calculate how long it will take for the particles to exit. So that's the clearance time. And then here's where things are no longer super intuitive. So you see that for a perfect orientational order, here we normalize it to 1. But actually, things are getting cleared faster if you add a little bit of disorder. So here where you see that around 0.8, we have a minimum. And then, of course, if you have more disorder, then things get trapped and don't exit this piece of tissue. But then what was really cool for us to see is that this minimum in this clearance time happens around 0.8, which is the actual measurement that we had from our airway data of the orientational order of the cells. So then this would really suggest that this disorder that we see is actually a feature in these tissues that allows them to be more robust, right? So then if you have an area where things are going nowhere, if you were perfectly aligned, they would just stay there forever. But then if you add disorder, then there's a path to move away from there. So this makes the airway very robust. So as I mentioned, we mapped a whole parameter space But this was kind of the highlight result. But then I hope that you saw like how this approach can be really valuable, even though our model might seem completely naive, and in a way it's abstracting a lot of the complexity. Then it can really help us think about how the function of this airway would work. So now in my group, what we're doing is continuing some of this work. And you can think in a biological system, as I mentioned at the beginning, there's lots of patterns, lots of parameters to measure. And then that's what we're doing because we want to really understand how behavior at the smallest length scale feeds into the other length scales. We are measuring all the way. An idea is like, okay, so how does the waveform of the actual cilia beat would influence their orientation in a cell? And then there's also other parameters that are not just spatial order, but there's a lot of temporal order in the systems as well that I will talk about in the second part of this talk. And so in the end, you can think of a whole parameter space to really map an image in the cells to try and understand how, if I change one little knob, how would that impact the other length scales that we have? And ultimately, how would that generate different flow patterns that we see in biology. And just so you get an idea of what are the diversity of flow patterns that we see, here you can see actually the flows. So cilia that I've been talking about are present in all organisms, basically across the tree of life. And here you can see the flows that are being generated by different arrays of cilia. And from a biological perspective, cilia are really highly conserved structures. So their ultrastructure and composition molecularly is pretty much the same. But you can see that really depending how you arrange them, you get radically different geometries and functions. So for example, here we have a starfish larvae. Here you see these flows that have tons of vorticity. And then in a way, this makes sense because if you are a starfish, then you want to trap particles because you're eating algae. So then you want to trap it there so you eat it efficiently. But this would be really bad if you're an airway and then you're trying to generate a unidirectional flow. So then we really think that in this case, it's all relies on how you arrange your array and then the emergent patterns that you see, then you can achieve different target behaviors. And so this is the approach that we're taking now in my group. And we're studying lots of different model systems because they all have different advantages and they allow us to access different length scales differentially, right? So because we do a lot of microscopy, it's not easy to image everything at every length scale. So then we kind of pick our model organism thinking about that. And then today I will tell you about some work that we are currently doing in Ciliated protist here at this length scale. And then to try and understand how temporal patterning of cilia arrays in the cells actually influences the function of the cilia array in the cells, which is swimming. So first, I will introduce you to ciliated protists. So I'm pretty sure that everyone is familiar with them because they are everywhere. So everywhere where you have water accumulating, every lake, after it rains, there's a puddle of water. Then if you take your foldscope, like Mariana was saying, you will find some of these little critters. So these are unicellular organisms. These are single cells. This is always something that was hard for me to grasp, but it's just one cell. And but they're pretty big when it comes to cells. They're 100 in the order of hundreds of microns to millimeters. Millimeter could be. And their feature is that they are completely covered with cilia. And they have all these interesting shapes, as you can see here. These are all ciliates, but they have a high diversity of cell shapes. And the ones that we're studying currently are highlighted here in green and blue. So that would be paramecium and didinium. And mostly we study them because they have nice features for us to image, but also because in the end, it's really hard to keep these ciliates in the lab. So then that's one of the challenges of actually studying them. Or from time to time, we go to Lake Geneva that you can see in this picture that is just kind of 2 blocks away from the lab. Collect water and then see what cool ciliates we find there. And here you can see actually some ciliates in action. So they're always moving, as you can see here. And this is how they would look. On the left, this is actually a video of paramecium that Scott Coyle took in some pond in Wisconsin. So these are actually like how the cells look in the wild. And now if we look at the organization of their cilia, as I mentioned, they're very interesting to us because they are completely covered in cilia, but they have different patterns. Here you can see paramecium is kind of this like baguette cell that has cilia that are organized in rows. And the dinium here, it's a round cell, and this is kind of a dream for a physicist because it only has two cilia arrays. Here you can see one at the anterior, which is close to this little dot that looks like a mouth, and then one at the posterior of the cell. So then it really has all the complexity of a cilia array, but kind of in the perfect geometry to understand what's going on with them. And as I mentioned, cilia arrays are not only patterned spatially, but also temporally. So here you can actually see a high-speed movie of what happens in the arrays of these cilia. You can already see this pattern traveling around the cell. This is what we call a metachronal wave. And metachronal waves, you might be familiar with them if you go to a football match all the time when you do the ola. So this is exactly a metachronal wave, right? So no one's moving. In this case, the cilia are not moving, but they're just like phase shifted, right? So so if one is kind of in this part of the beat, then the other one is just like a little bit further in the cycle. And that's how you see this pattern traveling around the cilia. So here you see a schematic of this, and then you can see the metachronal wave traveling around the posterior cilia array of the dinium. And as I mentioned, nothing is moving. It's just that the cilia are phase shifted in time. And just to give you an idea, these waves are traveling really fast. So at millimeter scales and millimeters per second speeds. And what we're trying to understand now is, well, part of the question is how do they form? Do they actually have any functional feature for this array? And just to get started, we're looking at what are their properties, meaning can we measure, for example, the wavelength of these waves and try to understand how these different parameters would change the function of the cilia array, which in this case is swimming. So I will show you some ongoing work in my group to try to understand this question in the dinium. Really, if we look at the cilia arrays and the metachronal waves in there, can we predict as a function of the parameters of the wave how this cell is going to swim? And here you can see a really nice immunofluorescence image of this cell. So the anterior, as I mentioned, is the side that has a mouth. So this cilia is actually a predator. It eats paramecium. So that's kind of nice for our lab setup because we have a whole ecosystem. So here's the anterior, and it has another cilia array at the posterior. And in the cells, we can observe for both arrays that there are these metachronal waves traveling. So here you can see kind of the profile view of the anterior wave in the array. And in the posterior, you can really image clearly because the cells have this behavior where they come down to the cover slip, they spin like a top, and then you can see this wave traveling around the cell. So this is mostly happening in the posterior arrays. And as I mentioned, ultimately what these arrays do is allow the cell to swim. And we can see that the cell actually can access a huge space of these different trajectories. So here you see the movie of cell swimming, and then on the right, you see the actual trajectories of every cell. You can see they do radically different things, at least just kind of on our first inspection. So what we're really trying to understand is if we look at a trajectory, like what are the cilia doing at any of these points that is allowing it to swim in this particular behavior? So then here you can see more zoomed in into a trajectory. So these are 45 seconds. And then as I mentioned, what we're trying to see now is how the motion changes in these different trajectories with respect to the cilia array. So here you can see the stretches that appear more smooth. So in this case, we see that the cell is kind of parallel to the surface, translates really fast. And we also see the cells go backward. So to go, the cilia reverse their direction, the cell swims backward, and then there's like some turning as well. So you can see that here. And as I mentioned, there's the rotation where we can really easily see what the cilia array is doing, where the cell just comes down to the surface and then spins. And then here you can see that it's not going anywhere. It's just spinning in place. So these are all the things that the cells can do. And at the moment, we're analyzing the cilia dynamics in these different swimming behaviors. So you can see that here the forward swimming to the left, both of the cilia arrays have metachronal waves. In backward swimming, it appears like it's the same, yet when you have to turn, you see that the wave disappears momentarily. And inside rotation, as we mentioned, you kind of see the cell go to the surface. And right now we're imaging these arrays, and then I'll show you some preliminary results of actually what are they doing when the cells are rotating. So what we found is actually that the posterior array has this super nicely coordinated metachronal wave, but then the anterior one is really disorganized. So this is the data showing this, right? So then here on the top, you can see the posterior cilia array, and here in the bottom, the anterior one. And then just from the movies, you can see that in the posterior array, you see this metachronal wave traveling. But if you look at the anterior, it just looks kind of like the cilia go everywhere. And if you make a chymograph from this data, in the middle here, you can really see, you see this perfect banding pattern, which just indicates metachronal coordination. But if you look at the anterior array, things just appear really disorganized. And if we actually look at the angular velocity, we can see that it's really constant, and this cell is just really rotating really nicely in place. So one of our hypotheses right now that we're actually testing with modeling is we think that in this case, the posterior array is kind of powering this rotation, while the anterior provides some drag such that the cell stays close to the surface. And if we actually look at what happens at the posterior array, as I mentioned, we're doing really careful measurements of the properties of the wave, because we want to now have a model and be able to encode these properties and see if we can understand how the cell swims. And then in a similar way to the airway work, now if we change these parameters, how can we, if we change these parameters, what will happen to the behavior? And we can also, what's nice in this case as well for the cells is that we could do a lot more experimental perturbations that, for example, the case of the mouse airway. But now, if we dive into the data, what's really interesting is that the properties of the beat of the cilia and the actual wave are highly conserved across several cells. So if we measure the beat frequency is cilia beat at 27, so 25 to 30 hertz. And what's interesting is that if we measure this across different cells, it's pretty much the same. And the wavelength is also similar. So here in this plot, you can see the measurements for different cells. So we see that the wavelength is 22 microns. But it's also pretty consistent across different cells. And so similarly, we can look at how many nodes the wave has. So then it has around 17 of these nodes. And this is really interesting because, well, the cells can have variations in their size. But here you can see that the ratio of the number of nodes divided by the radius of the cell is pretty constant. So then there seems to be some target kind of linear density of nodes or something like that. So this is for average measurements. But what we started to see is if we actually look closely, we see that if we look at periods of time, this is not constant over a whole cell. So here I will play this video. And then you can see that on the left side of the cell, it has a wavelength of 21 microns. On the right side of the cell, 37. So then waves are kind of bigger, the wavelength. And we think that this is kind of similar to what has been seen for other swimmers, where you want to steer or change direction. Then you kind of need to, you have these glitches in the coordination. So we think something similar might be happening here, where maybe the cell is going to go somewhere else or do some other swimming behavior. And that's why we see that there are these differences or glitches, spatial glitches in the coordination. So that's what we're currently investigating further, doing more measurements in the cells, doing measurements in also the other configurations where you see the cell swim kind of faster. And again, we're with our collaborators, we're building a model to very similar to the airway work, try to understand how would this metachronal wave properties give rise to the swimming and of course the flows. So we can always image the flows that are being generated by this arrays of cilia. So here you can see an experiment where we're doing this, and we're trying to map if we can see signatures, for example, of the wavelength or the spatial organization that we measured in the structure of the flow to also understand not only the swimming, but then of course this has to be mediated through the flow. So here you can see just one of those such experiments. But so this is what we're doing in terms of studies that we're doing in the Dinium. But as Mariana also mentioned at the beginning, at kind of during grad school, I was super interested in the cytoskeleton and how the geometry of these arrays gives rise to different physical forces. So I was happy that after studying cilia and thinking about cilia still for a while, I kind of find a way back to studying the cytoskeleton, because we started to look at this other cytoskeleton-like proteins that are in this protist, like paramecium, that allow them to execute unique behaviors. So when I say unique behaviors, this is what I mean. So here on the left, before I play the video, I'll explain it a bit. Here you have the dinium. This is the round cell held by a pipette. And then here you have paramecium, that the dinium is paramecium. And then you will see what happens. So basically, the dinium is going to engulf a cell that is at least twice its length. So to be able to do this, it needs to undergo this extreme shape change. Yet, as I mentioned, it relies completely on the organization of cilia to swim. So then this is kind of a competing function, right? So it needs to preserve the cilia organization, so it can't change a lot the cortex, but it needs to deform extremely. And similar, here you will see a movie from Lacrimeria, again taking mesoscopic coil. And then here you can see that the cell extends its neck to grab some prey. And as well, it undergoes this crazy extensile deformations. So we're going back to the cytoskeleton and thinking about which proteins or which polymeric assemblies could actually mediate such behaviors that we see in the cells. And the one protein that we've been studying and focusing on is centrin. So centrin is really well known and most well known in the context of its presence at basal bodies and centrosomes. So it's really important in some cases for ciliogenesis and for duplication of centrosomes. But here you see it in this protist forming this crazy arrays with a highly stereotypical geometry. So here you can see a little inset from paramecium. This almost looks like a mesh-like network. And this is what is known to underlie the actual shape changes observed, for example, in the cell. So what we're studying now is also how the geometry of this array actually could give rise to cellular scale deformations upon deformations of kind of the filaments that form this array. But we know a lot less than what we know for actin cytoskeleton, for example. So this is a really interesting and exciting problem. And here you can see, you can see better the geometry of this array. So here in magenta, you see again centrin. And here you see the cilia as well. So you can see that the cilia pattern perhaps also is patterned by this arrays of centrin in this case. And now what's really nice is that we can also do expansion microscopy and look at really all the detail of the geometry of the cellular arrays of centrin in the cells, and do some measurements of this geometry and try to understand how deformations of this network would give rise to deformations of the cells in this case. So this is another problem that I was really excited to go back kind of to studying as cytoskeletal mechanics. And as I mentioned, there's lots of interesting questions coming up here because just there's not a lot of work been done in recent years in this field. So then we don't know even how would this assemble into a mesh, how does centrin assemble into a mesh, what are the mechanical properties of these more networks, and how do they kind of balance these two competing needs where you have to undergo this really big deformation without changing the pattern. So there might be different mechanisms at play, different from polymerization, for example, in the case of actin. So we're thinking about the centrin assemblies more like metamaterials, perhaps, where kind of you can think of these origami sheets that are cut in a pattern, and then it's just a sheet that is folded, but then you unfold it, and then it increases its length greatly. So this is kind of a new direction of us that we are exploring for us, that we are exploring in the cells. And finally, because we don't stop to find interesting problems when we looked at these ciliates, as I mentioned to you, for us, it's nice to have both paramecium and didinium in culture because to keep didinium alive, we have to feed it paramecium. And you can see in this electron microscopy image kind of how didinium captures paramecium. And now we're trying to understand how it finds it. Again, going back to looking at cell trajectories of swimming of didinium, can we understand how it searches for its prey? So we're also doing this type of behavioral imaging and analysis, where here I will play this movie and you will see something interesting happen in these green circles. So here you have didinium and it will just capture the paramecium on the left. And then I think the second one will come on the right. Oh, that one escaped. But there you go. We are looking into how didinium changes its swimming behavior in response to seeing paramecium, or can it see paramecium, or is it always there? So this hopefully gave you a good overview of the types of questions and types of approaches that we have to really try to connect these scales and try to connect really the cell biology, the molecules with the biophysics to understand the macroscopic behavior of systems. And then, as I mentioned, we're currently studying a lot of ciliates because we continue to find interesting questions exploring the cells. And with that, I would like to thank you for listening and thank to all the members of my lab, especially Katerina Korkula, who is the one who's doing all these careful measurements in didinium metachronal waves. And thank you.
That was a great talk, Guillermina. Thank you for such a nice presentation. I think we were all in awe with with the videos and seeing all the incredible biology, mechanics, etc. So we have just very few minutes, so maybe a couple of questions. Is there someone from the audience that would like to ask? Some of you can ask in Spanish if you wish. Or in English. I have one question. So, well, I have many questions, but what's it's such a complex topic. So I remember when I read your paper on the lungs, thinking this is one of the many things, right? You also have like motion from respiration, you have the vascular flow. So I was, studying pathogens in vasculature. So you have all of these different things coming together. how difficult is it to model something like that, right? I remember when I started modeling, people would tell me, if you assume biology, it's already much more complicated. So how has it been for you, all your path to building all this, knowledge?
Well, I think I guess that's the art of doing this in a way, right? It's an art to really trying to understand which parameters matter and which not. So I always think about all this microscopy data, and we could measure a ton of things all the time.
Yeah.
But it really goes back to thinking about for the behavior or the problem you're trying to understand physically, what matters. And try to I think it's really nice then to go back to this soft matter literature, where a lot of you can see already, okay, how would you measure order in the system? What order is important for this type of thing? But
Yeah.
then, of course, you can't lose sight of the biology. So I think that's why kind of one really important thing that this is a super interdisciplinary problem, right? It's not just my group and me. Like I have tons of collaborators that keep me grounded all the time. And like you really need kind of this exchange to make this happen in a balanced way, I guess.
There's a question from Esteban in the chat, which is great talk, Guillermina. I was wondering if there are any interesting genetic variants of cilia to maybe change the behavior, control the movement of these organisms.
Oh my God, there's so, okay, so in this ciliate literature, what's really interesting is that these are cells that people studied in like the 50s, 60s, 70s, 80s, and that's it. And they have all these super cool descriptions of, for example, in Paramecium, there's this idea of structural inheritance that like people can actually, just do a cut, kind of invert the cilia, something like that. And this is this error in the patterning is propagated through generations of cells. So somehow you have all these patterning variants that you can create physically. And then also there's some molecular perturbations that now we're doing also in Paramecium. I didn't talk about this, but we're looking at the metachronal waves there, and there you can, knock down lots of proteins that give you different array phenotypes, and it's a lot easier than, for example, mice. So there's a ton to explore there.
And maybe now we're in the hour, but there's so many questions we would have. But maybe whoever feels interested to continue the discussion with Guillermina, you can send her an email. One last question, just to wrap up, which is, unless there's something from the audience, from me would be, how has your path been? You went from Mexico to the US, and now you're a PI in Switzerland. How has it been for you to become a PI? You're a young PI, right? How has this
Well,
path
I look younger than
you?
what I am, but no, I think this connects nicely kind of with this part that I didn't touch about my heritage and how I approach life. Is very much Mexican. This is the Mexican way of living where you, if you've ever been in Mexico City, you walk out the door in the morning and you expect nothing to work. Nothing works. Like, somehow you have to improvise at every step and be really good at like figuring things out on the go. So I feel like this has been my path, completely random. Like I never saw a path, I went for, I was like, oh, I love math, I'll study physics. Why not? And then I was like, oh, like I need to do a thesis. There was this one lab that had an opening, I joined it. And then I was like, oh, I love biophysics. I'll just Google biophysics PhDs, apply. And then I was like, oh, now because life stuff, I need to move to the Bay Area. Okay, these two labs seem fine. I interviewed in a bunch. This gave me a position. And like, honestly, moving to Switzerland was like, oh, okay, like there's this opportunity. Sure, it sounds good. But
which is a fantastic lab. It's a fantastic lab and a fantastic institute. So
but I think this very much speaks to kind of like, how you just hope for the best, go with whatever gets thrown at you, and then kind of try to see kind of like, it will work out in the end. I think this is kind of something very much for like, for my experience, how I grew up, and like, my family and everything. And I guess the other aspect is always like wherever I go, I know like I will need to find a community, build a community. So that's
Yeah.
another kind of very Mexican thing in a way. Like I know I cannot do anything by myself. I need like the support of everyone and everything. So I think wherever I go, I really try really hard to find people. It's not obvious where they are, but then
Yeah.
this is kind of also a skill I think you have as you know, growing up in a giant family.
Mhm.
You just know you have to find your people in the way, and kind of you just go wherever and find your people.
Yeah, that's a great view. So actually, Holly Aaron says, Guillermina, I love the way you see the world, looking at all these patterns across vast scales. Like this is so fascinating. Congratulations and thank you for sharing your work.
Oh, thank you. Thanks for
Um,
the invitation. I really enjoyed giving this talk, and
no, thank
you.
you. And you were the last speaker of this series. It was a wonderful series and I think a wonderful ending. So thanks to everyone who made it possible and to you, of course.